Good morning, everyone. My name is Jedric, and together with Lucas and KaiXiang, we will be presenting on the human T cell leukemia virus type 1, more commonly known as HTLV1. In this presentation, we will mainly focus on how HTLV1 infects a host cell and goes through its viral life cycle. We will also briefly discuss how infection can affect the human body. To begin, HTLV1 is a virus that uses RNA as its genetic material. When HTLV1 infects a cell, its RNA is converted into DNA. This viral DNA is then inserted into the host cell's own DNA. Once this occurs, the viral genetic material can remain within the infected cell and can later be used to produce more viral RNA and proteins. These components can eventually form new virus particles, which may then spread to other cells. An important characteristic of HTLV1 is that it causes a chronic and lifelong infection, which is closely related to its ability to integrate into the host cell genome. This diagram gives an overview of the HTLV1 life cycle. We have divided the process into 10 main stages, beginning with adsorption, followed by signaling and penetration. After HTLV1 enters the cell, it undergoes uncoating, nuclear targeting, and integration. Gene expression, genome replication, and, in some cases, viral latency. Finally, new virus particles undergo assembly, maturation, and release. We will now go through each stage individually. The first stage is adsorption, or attachment. HTLV1 mainly targets CD4 positive T cells, although other immune cells may also be infected. The virus contains a surface glycoprotein called GP46, which recognizes and binds to molecules on the target cell surface. Attachment occurs through several host cell factors. Firstly, HSPG captures HTLV1 and brings it close to the cell surface. Next, NRP1 strengthens the attachment. Finally, GLUT1 helps facilitate the next stage of viral entry. Another important feature is that HTLV1 commonly spreads through direct cell-to-cell transmission, rather than relying mainly on free viral particles. Once HTLV1 has attached to the cell, the next stage is signaling. Binding of GP46 to the host cell receptors does more than simply hold the virus in place. It also activates signaling pathways inside the CD4 positive T cell. Some important host proteins involved are PI3K, SRC, and RAC1. PI3K is involved in signaling pathways related to cell survival and membrane changes. SRC helps transmit signals from the receptor complex into the cell, while RAC1 helps control changes in the actin cytoskeleton. The combined activation of PI3K, SRC, and RAC1 results in actin cytoskeleton remodeling. The actin cytoskeleton helps maintain the shape and organization of the cell. When it is remodeled, the structure and organization of the cell also change. These changes help prepare the CD4 positive T cell for the HTLV1 entry and viral transfer. So, HTLV1 does not simply attach to the cell, it also modifies the host cell signaling and structure to support infection. After attachment and signaling, the next stage is penetration. During this stage, another envelope glycoprotein called GP21 becomes especially important. GP21 undergoes structural changes that bring the viral envelope and the host cell membrane close together. This allows membrane fusion to occur. Once the two membranes fuse, the viral core, containing the viral RNA genome and essential enzymes, can enter the cytoplasm. As shown in our diagram, endocytosis has also been reported as another possible entry pathway in certain host cells. However, membrane fusion is considered the main mechanism of viral entry. Once the virus has successfully entered the cytoplasm, its components are positioned to begin the next stages of the viral replication cycle.
After the viral core enters the cytoplasm, the fourth stage is uncoating. During this process, the viral capsid gradually disassembles and releases the components contained inside it. One of these components is the viral RNA genome. This RNA contains the genetic information needed for the following stages of the viral life cycle. The virus also releases reverse transcriptase. Reverse transcriptase converts the single-stranded viral RNA into double-stranded viral DNA. Other viral enzymes, including integrase and protease, also have important roles later in the life cycle. Therefore, uncoating prepares the virus for reverse transcription and the later integration of viral DNA into the host genome. The fifth stage is nuclear targeting and integration. Reverse transcriptase first converts the viral RNA into double-stranded viral DNA. This newly synthesized viral DNA then associates with viral proteins, including integrase, form pre-integration complex, also known as the PIC. The PIC is transported through the cytoplasm and enters the host cell nucleus. Once inside the nucleus, the viral enzyme integrase inserts the viral DNA into the host cell's chromosomal DNA. After integration, the viral DNA is known as a provirus. This is an important stage because the viral genetic material has now become part of the host cell genome. The provirus can remain inside the infected cell and be maintained when the host cell divides. It can later act as a template for viral gene expression and production of new virus particles. The sixth stage is gene expression. At this stage, the host cell transcribes the integrated provirus into viral RNA. Different forms of RNA are produced. Spliced viral mRNA converts regulatory proteins. Partially spliced RNA can encode envelope proteins such as GP46 and GP21. Unspliced viral RNA can encode structural proteins and viral enzymes. The host cell ribosomes then translate these RNA molecules into viral proteins. So essentially, HTLV1 uses the host cell's own machinery to manufacture the components it needs. The seventh stage is genome replication. Unlike many RNA viruses, HTLV1 does not directly copy RNA into more RNA. Instead, the integrated proviral DNA acts as the template. The host cell's transcription machinery reads the provirus and produces full-length viral RNA. Some of these full-length RNA molecules become the genomes that will later be packaged into newly formed virons. Other full-length RNA molecules may also be used in the production of viral structural proteins. So, genome replication in HTLV1 is essentially the production of new full-length viral RNA from the integrated proviral DNA.
The H stage is viral latency. After integration, the provirus does not always remain fully active. Instead, it may enter a state where it is inactive or only partially active. During latency, there is little to no viral transcription, so very little RNA and few viral proteins are produced. Reduced viral gene expression can help the infected cell avoid detection by the human system. This allows HTLV1 to remain within the host for many years. However, latency is not a compulsory stage that every virus must pass through before assembly. Instead, it is an alternative state of the integrated provirus. The provirus may remain relatively silent or viral gene expression may become active again. Therefore, latency acts as a survival strategy that contributes to the lifelong persistence of HTLV1. When viral gene expression is active and enough viral components have been produced, the next stage is virion assembly and maturation. Full-length genomic RNA and viral proteins move towards the plasma membrane. At the same time, viral envelope proteins are inserted into the host cell membrane. These components gather together and form an immature virion. However, the immature virion is not yet fully infectious. The viral enzyme protease cleaves viral polyproteins into their functional forms. This causes the internal components of the particle to reorganize. After this processing, the immature virion becomes a mature infectious virion. So, assembly forms the new particle while maturation makes the particle infectious. The final stage is release, or shedding. The mature virion pushes outward from the host cell plasma membrane and leaves the cell through a process called budding. During budding, the virus acquires a lipid envelope from the host cell membrane. This envelope contains viral glycoproteins such as GP46 and GP21, which will later help the virus infect another target cell. After release, HTLV1 commonly spreads to nearby cells through direct cell-to-cell contact, including through a virological synapse. Once it reaches another target cell, the cycle begins again. After understanding the life cycle, we can also look at the effects of HTLV1 on the human body. HTLV1 mainly affects CD4 positive T lymphocytes and can cause persistent changes in immune cell division. It may lead to abnormal T cell proliferation and chronic immune activation. In a small proportion of infected individuals, HTLV1 can cause adult T cell leukemia, lymphoma, or ATLL. It can also cause HTLV1 associated myelopathy or tropical spastic paraparesis, also known as HAM or TSP, which can cause progressive weakness and stiffness in the lower limbs. HTLV1 is also associated with inflammatory conditions such as uveitis and infective dermatitis. However, most infected individuals remain asymptomatic even though infections can persist for life. To conclude, HTLV1 has a complex life cycle involving attachment, signaling, entry, reverse transcription, integration, gene expression, genome production, assembly, and release. A particularly important feature is its ability to integrate into the host genome and remain latent, which contributes to lifelong infection. Its efficient cell-to-cell transmissions also helps it spread between immune cells. That concludes our presentation on human T-cell leukemia, virus type 1. These are our references.
Thank you.
Thank you for listening.